peak intensities again? Like,
Yes.
So if you can go to that thing, so this is a common platform for all types of data analysis. So for mass spectrometry, we go for peak intensities or the spectra. For NMR files or the NMR data, we go for that kind of thing, spectral
Spectrum.
beams. Okay, for NMR files and all those things, and any other, like if you go for FTIR and all those things, you can go for concentrations. But it's okay, like whatever the biostatistics you perform is good to go. Any doubts in this? Okay,
Yeah.
great. Proceed.
Yeah. So after you upload the file, so there will be this first page. Just bear with us. Yeah, so this is the data processing information. So here you can see what are the how many samples were there and how many features or peaks. I mean, in this case, protein IDs are there. This the first thing here, 8 samples by 1953 peaks. So this you can also check directly from the Excel file if you just check how many proteins are there and how many samples are there. So you might be asked this. So what is the matrix like 1953 protein IDs or proteins and 8 samples? So there are two groups detected, as you saw on the Excel file. There were control and COPD groups, so two distinct groups, and a total of 11.6% missing values were here, and that is 16.58. So 11.6% missing values were detected. So this is the preliminary information that is given to us, and we need to, and based on our preference or what we need, we can then further process this data. So first we do the missing value imputation. So as you saw, there are around 11.6% missing values in this data. So first we'll impute the missing value. So in a generalized rule, we generally remove features, that is the proteins which are there in the first column you saw in the Excel. So on the proteins which have more than 30% missing values across all the samples, we will remove them. And so if you increase this percentage to 50, you are actually like making it less strict. So the more the percentage, if you reduce it, you are making it more strict. So in this case, we are removing protein features which have more than 30% missing values. So first we'll do here 30%, and then we do like what are the missing values present. We do the missing value imputation by k-nearest neighbor. So like it looks into the cells adjacent to the missing value, which so for example, on a particular cell, we have a missing value. It will look into the adjacent cells, and based upon a KNN algorithm, it will like impute the missing values. So do this and then process. So in questions, you might be given a different values like 50% missing value, 20%. So based upon that, you need to do this. So this is not a very stringent criteria, but on a rule of thumb, in our lab, we use 30% missing values and KNN feature-wise imputation. So on this page, do nothing, just proceed. And now we need to normalize the data. So before that, when you did the missing value imputation, so there might be some proteins which had more than 30% missing value, and so they might be eliminated from the Excel file. So if we want to see how many proteins are there after the missing value imputation, so we can go to download here on the left hand side, and we can download this one, this raw data. This is the one which we uploaded, and the data process CSV. This one we need to download to check if how many proteins are there now. So if we save this, yeah, yeah. So this is the process data after the missing value imputation. So if I see the number, so it's around on the here you can see the count is 1482. On the initially it was 1953 features were there. After the missing value imputation, the count got reduced, and now it is 1482. Yeah, so this is okay. Yeah, so just let me go through this one more time. So do nothing here, just proceed. Yeah. So I hope you have understood the missing value imputation thing. So now we need to do the data normalization. So normalization is generally used to like minimize the effects across the runs or the variations across the run. So we do normalization. So we usually do median normalization as well as log transformation. So why we do log transformation? For example, we are dealing with intensities here, intensities of different proteins across the different samples. So for example, on a hypothetical sense, if I say one protein has an intensity 100 and one has an intensity of 1 lakh, how can you plot that in one axis? So when you log transform that, it gets reduced to log 100 is 2 and log 1 lakh will be, I guess, 4 or 5, something like that. Yeah, so it just reduces the scale variation and you can fit it into one scale. So you do here median normalization, log transformation, and then you click normalize and then view result. So on the view result part, you go to sample view and then you see here on the right hand side. So this was before the normalization here, and after the normalization, it is like perfectly fitting the box plots, and you can see this is the curve. So based upon different samples, so here you can see like this one curve, there is one more small peak, or in case of other data, you can see one clear Gaussian distribution. So that this is totally dependent upon which sample are you using, and it differs widely. So this is the nomination has been done, and so then you can proceed next. So first we do correlation heat maps to check if there is any outlier in the sample. So here there were 3 control and 5 COPD or 5 disease groups. So we need to check first if there are any samples that are outliers. So just a minute. Yeah. So we choose this dimension, change it here to, it is by default it gives features, but we change it here to samples. And then we do the Pearson or correlation. So there are 2 types, Pearson and Spearman. So they, if you go into statistical analysis, Pearson measures the relationship between 2 variables which have a linear relationship, and Spearman measure between variables having a monotonic relationship. We do not need that as of now. So samples here, Pearson, and nothing, just click submit here. Yeah. So you can see this is a correlation heat map. So you can see there are no outliers, like, and the minimum correlation coefficient on the right hand side bar, you can see it's 0.9. So very good correlation coefficient. You can see, so, and these are the samples on the right hand side, and this is the same thing. So if there were any outliers in this, so you will get a very distinct or definite change in color here, and you will be easily able to distinguish the outlier, and we need to remove the outlier from that case. So in the data file I provided, so there are no outliers. So you can also download this file from here, from this PNG section, or you can also change the color contrast if you like. So you can do it red, green. These are like some of the parameters you can play with. So first, after we did the correlation heat map, then we look at the PCA plot, which is the unsupervised clustering algorithm. So we go to PCA and we go to the 2D scores plot. So here you can see, so here it has been, the green one is the normal and the red one is the COPD, and we are displaying the 95% confidence regions. So here you can see there are separation, but 2-3 samples are like on the same, like on the same area, and they are not like totally clustered, the 2 groups ideally. So if the samples are totally like it is control and disease, they're totally distinct, and if the samples have been processed with very good hand, and if the person who is doing the experiment is really experienced and has good hands, so most of the time we get a clear clear clustering. But in some cases, if there are tissues and generally biological samples, you also might not get a clear clustering. And so here you can see there is no outlier as well. So if there would have been any outlier here, you would have seen 1 or 2 data points like this red or green outside this 95% confidence interval, that is the red or green sphere which you see here. So if outlier, they would come on any of the side outside this sphere. So no outlier as well here. So now we can also look at the PLSDA. So one difference between PCA and PLSDA is PCA is principal component analysis. It is an unsupervised learning. So unsupervised means we just gave this the input data, like the peak intensities we are having, but we did not mention that this protein and this peak intensity is belonging to the normal, or this is belonging to the like COPD, that is a disease. We just gave the intensity.
Okay, just a minute. We have 2 questions. One from Dr. Shivalakshmi. Yes, Dr. Shivalakshmi.
Okay.
Sir, if there is any outlier, like color will be different, or how it will be?
Color won't be different. For example, see, just give me the sample names. So you see here DC3, DC2. So if DC2 would have been an outlier, this point would have been outside this sphere. So
Not
whichever
in
point.
Not in PCA, sir, in heat map.
heat map. So heat map, it will be different color. Yeah.
Different color.
Different color. You will be clearly able to distinguish.
Oh, thank you.
Okay. And one more question, I guess, from Sana.
Actually, I'm getting a very different kind of plot from what you have shown. So I just I'll just share my screen, and can you explain that?
Yeah. Sure.
Are you able to see my screen now?
Yes.
Yeah, so I think in your scope plot, the green part was below the part.
No. Yeah, this is one thing which we faced also. So usually this happens if the naming is somewhere different. So you can see if closely look at your plot and the plot I am getting. So it is essentially just a mirror image type of thing.
Yes, correct.
Yeah. So this doesn't isn't different on that sense. So for example, if we name the samples different in the Excel sheet, sometimes this happens, but this isn't an issue as well. This is just a mirror image. You see again, just the same. And the
Okay, fine.
PC values are also same. PC1 and your PC2 is 15.8, and if you just scroll down, we can see 28.2.
28.2.
Yeah, here it is also the same. So this is actually the same scores plot, but sometimes it gets flipped because of some naming or thing.
Okay. And what is the difference like PC1 and PC2?
So these are the first two components. So PC is usually a dimensionality reduction thing. So like there are many dimensions, so but we reduce the data to actually reducing it to the first two principal components. So PC1 and PC2. So we can also look further, but yeah.
So PCA is a principal component analysis, is a form of doing a supervised clustering. Like when you have two groups, unsupervised clustering, when you have two groups, you can mute yourself. When you have two groups, one is the control and one of the disease, you feed it to the system and tell it like, on the basis of best classifiers, classify my these two groups so that I can segregate these two cohort. We are not telling that this is a control and this is a disease. We are just giving the groups based on the eigenvalue calculations with the straight line. That is the dimensionality reduction. They calculate the algorithm and best of 5 is taken. The most significant one, PC1, PC2, PC3, PC4, PC5. The most significant one which we see are PC1 and PC2. If you are doing a supervised, like if you are subjecting the algorithm that this is my control and this is my disease, these are my two groups. On the basis of that, you select out the best classifying clusters. That becomes a PLSD. Okay, that is the PLSDA with the linear discriminant. Similarly, you can go for orthogonal PLSDA also and bigonal PLSDA also. So PC is a form of PC means principal component analysis, is a form of unsupervised, which will give you a data how your data or the dimensionality or the reduction looks like. And generally the PC1 and the PC2 components should have a difference of 30 to 20%, because then only you can understand that your data is segregating or not.
Okay, thank you, sir.
Okay.
Yeah, thank you. Just let me share it again. Yeah, so as Dr. Orko just now mentioned, so PCA is unsupervised, and if we go into PLSDA, so on the while doing the PLSDA, we are telling the system that we have two different groups, control and the disease, and how best you can now classify the two groups. So here you can see the clustering is very good, and the two groups are totally segregated. So because we have already provided the algorithm that this is sample and this is the control sample and this is the disease sample. So this is a PLSDA. And also in some cases you might be asked about this important features thing. You go here, important features, and you might be asked what are the top 10 features or top 15 features. You can change the number here. For example, you can go to top 10 here. So we'll get the top 10 features. This is VIP, this is variable importance projections, VIP scores. So you might be also asked this. So what are the top 10 important features in terms of VIP scores? So this is the PC and PLSDA. So next we go into heat map. So we click heat map from the left column here. So this you do not need to change anything. You can change the color because I like the data to be visualized in red and green. So you can change the color. And here you can use, for example, you want to see what are the top 10 features here. So I use top 10 and then I do not cluster, and we can change it to here features on this thing, and we click submit. So if we go down now, here you can see there are two different clusters. Like on the left hand side, this is the COPD, 5 groups, 5 COPD samples have been shown. On the right, 3 control, and you can see the top 10 protein features which are there and how are their expression levels across the different samples. So here red, red means the protein is significantly upregulated, that is it has a higher fold change, and on the green means here on the it is also already defined in the axis here. So the green means it is down regulated or having a lower fold change. Or you can also change this if you do here samples, not features, then you can see this will be at the same time. So all the upregulated proteins will be shown as a group, and all the downregulated proteins will be shown as a group. So this is based upon what you want to see. And on this thing, the top 10 features or top 25 features, for example, if I do top 25 features, this thing will, for example, let's try with 50 and do here features. So this type of figures you will typically see in a paper. So yeah, this is the top 50 protein features are here, and what are their expression levels across the different samples. So this is the heat map. So you can also download this and use as a publication figure. Next we go to the volcano plot. So which, let me open it first. Yeah. So volcano plot usually takes into account 2 parameters. What is the fold change? So I think we can go to fold change first also. Let's go to fold change here, and we can give any threshold like fold. And here you can see direction of comparison is COPD by normal. So we are taking the disease divided by the control. And we want to see, for example, which protein features have a 2-fold or anything. We can change the fold change to 1.5 also, because sometimes we do not. So the fold change threshold, if we reduce it, the number of proteins we get will be higher. And if we make it more stringent, like 2-fold change, 3-fold change, the number of proteins will be reduced much. So if for example, I give here 1.5 fold change, and we see which proteins are 1.5 times they are upregulated in the COPD disease group as compared to normal. So we click here submit. So you see here 48 proteins have a fold change of more than 1.5 in COPD group compared to normal, and 63 are downregulated. So they have fold change of less than 1.5. So, and if you can see, go to the volcano plot, because we use the volcano plot directly. So this takes into account both your fold change as well as the p-value. We give a p-value cutoff here. So the fold change, as per the previous screen, we gave 1.5. We are doing the direction of comparison. Disease divided by the control, and we usually set the p-value threshold cutoff to be 0.05. So that is or we can also change it to 0.01. That will make it much more stringent, but 0.005 is the usual one. So group variance, we change this to unequal, and then you click the submit button. So here you can see based on this p-value threshold 0.5 which we gave and the fold change of 1.5, we are getting 14 significantly upregulated proteins and 13 significantly downregulated proteins. So just want to ask if there is any question up until now. So because this step is important. I guess no questions. Yeah. So this thing is very important. So here you can give any threshold criteria. So if I give here 2 fold change and keep the other parameters, then we will see this number is getting much more less. So there are only 1 downregulated protein and 5 compared to there were 13 and 14, I guess, on the previous one. So if I, for example, keep this 1.5 and we want to look into which proteins are significantly upregulated and downregulated and what is their fold change. So this is the volcano plot. You can download this plot, and also you can see what is the list of proteins and what is their exact fold change. So you click on this here. So this is the feature details table. You can download this whole thing as an Excel file. Yeah. So this Excel file gets downloaded. So here you can, FC is the fold change, and this log to FC is the like logarithmic version. But we want to look at the absolute fold change, which we give the criteria to be 1.5. So first we sort this data. We click this column here, data Z2A from the highest to lowest. We sorted this data. We see there's one protein. This one is 39 fold change, very highly upregulated. And from below this is the 1.5. So proteins above this one are, let me color this first. So all these proteins are upregulated here. So based on the heat map, we gave it red, and all the proteins having fold change of less than 1.5, they're downregulated. So we're giving them, for example, green. Yeah. So here you can look into your data, like which proteins are highly upregulated, which proteins are downregulated. You can play with this. And also what is the p-value criteria. So all these are less than 0.05 because we set the criteria. It won't show any proteins having higher p-values. So if now we want to look into which proteins are these. So we can go to ID mapping, we can go into UniProt, we can input this protein IDs, and we can see which proteins are these actually, which genes they belong to. Or we can go to pathway analysis in Reactome. So let me open Reactome first. Yeah. So just Google Reactome and open this reactome.org. Go to analysis tools. Yeah. And for example, here you want to see which pathways are being affected by your this disease condition. For in this case, this is COPD. So we just copy all the protein IDs. So here are around 27 proteins, the count, and we paste this into Reactome. We click on continue, and so here it is project to human. So if, for example, you are doing this on, actually this data is also in rat. So this study was done on rat, but we want to see the human analogs, like how this, like rat proteins and what are the human analogs. So we want to project to human, and we can also do the include interactors if you want to see what are the closest interactors of this protein, for example, in a biochemical pathway. But for here we don't want to see the interactors. We just want to see what are the proteins, how these proteins and what are the pathways they belong to. So we click on analyze. So here you can see the pathway names, entities found. There are a lot of information here. So we can visualize this data, not visualize, we can see these data on a much easier version. We go to downloads here, and on the pathway analysis result, we want to download an Excel file in CSV format. So click on CSV. So an Excel file has been downloaded. Just let that open. Yeah. So you can see what are these are the pathway identifiers, the first column, then what is the name of the pathway. So we're interested with the name. And this is the third column is the entities found. For example, how many proteins in the list which we provided, the 27, the count, if you remember. So among the 27 proteins, how many proteins are mapping to this pathway, erythrocyte taking up oxygen. And for example, this evaporia formation, again, cytoprotection by HMOX1, here 2 proteins are being mapped from our list. And on the right hand, on the D column, you can see what are the total number of proteins that are mapped to the pathway. So out of 8 proteins in this first pathway, 3 proteins from our study are mapping. So we can say this is we can say confidently that this pathway might be very important, very crucial in our study. So entities found, we can just check what are the pathways on the basis of highest number of entities. So if we sort this data here, yeah. So on this pathway, 5 proteins from our study are mapped, but this is metabolism, protein metabolism. For example, transport of small molecules here, 4 proteins have been mapped. Then again, erythrocyte taking up oxygen we saw earlier also. And this is the total number of proteins in that pathway. So entity ratio, this is just the dividing. And what is the p-value? For example, somewhere there. Yeah. So now we can also sort this table based on the p-value. So sorry, in this case we need to do the lowest to the highest, A to Z. Yeah. So we can also check the pathways based on the p-value. For example, we want to delete we are not interested in the pathway which are having a p-value of higher than 0.5. So this is the pathway 0.05 and this is having 0.06. So we aren't interested in pathways having p-value of which is higher than 0.5. So we can just delete all these. Yeah. So there are around total 18 pathways which are having p-value of 0.05 or less, as you can see here. And this is FDR we don't need. And on the basis of reaction find, so you might be asked different type of question that you need to just play with this Excel sheet. So you might be asked what are the total number of pathways having p-value of less than, for example, 0.01. So you just need to say it is 6. Or you can be asked what is the number of pathways, which pathways having reaction more than 2, more than 5. So you just need to, whatever question you are being asked, you just need to play with the parameters, reactions ratio. And one more thing, you can also be asked, for example, mapped entities. So which proteins were mapped into pathway, for example, this one or this one, ECM. So extracellular matrix organization, we are selecting this row. So you might be asked what are the proteins that are being mapped into the pathway extracellular matrix organization. So you need to go here. These 3 proteins are there. You can just type all of these proteins in UniProt, or you can just mention their protein IDs and you can say, okay, these 3 proteins are mapped into the pathway extracellular signaling matrix. Or you might also be asked between these 2 pathways, extracellular matrix organization, as well as this oxygen carbon dioxide exchange in erythrocytes, if there are any proteins that are common in these 2 pathways. So you can just note down what are the proteins in this pathway from here, mapped entities, and you can also note down the proteins that are here in this pathway from the mapped entities. And you can just say, okay, one protein is common between these 2 pathways, so 2 proteins is common between these 2 pathways. So this is very easy. So this is it for Reactome, and I think this will help you with the assignments. So if there are any specific questions or you want to do it right now, you can just let me know. That's when I'm stopping my screen. Someone has a question, I guess, over there.
Yes, Sana.
Actually, I wanted to know that when in the reactome analysis that you have entities found and entities mapped and total.
Yeah.
What is the difference between total and found?
Just let me go to the thing again. Yeah, so this found thing is how many proteins are there in our in our list, and that has been mapped.
Click on that, click on that, click on that. Just see the topic.
Yeah.
Okay, so and just at the near that entities.
This is total.
Okay, so if you want to see, one is the pathway. When you go to the reactome, you see sub pathways. Okay, there is a main pathway, then there is a sub pathway. So that particular compound is there. Please show the sheet. Yeah, so that particular compound, like if you can see, at the right hand side, you can see map to, okay, P00915, P6. These 3 entities are mapped, okay, in that particular pathway. And when found in the reaction, like RS12476668, what all other reactions are there, okay, these are also parallelly found in those reactions. So now you are clear with found and mapped, correct?
Yes, sir.
Okay, so at the right hand side, you will be getting everything.
And
Which
what do you
all
mean by
pathways they are.
total entities total?
Total entities means total number of reactions it is mapped to.
Okay.
So just click on submitted. Submitted column. No. For yes. Just click on that at the right hand side. Found reaction and identifiers. It should be 18. Somewhere it should be 18. Sorry, 8. So Sana, you can play around this. Everything is informed in this only, in the submitted, mapped and found. Like if that particular protein is mapped in multiple pathways, or it is occurring in a particular pathway, those all scores and values are given. Okay.
Okay,
Yeah,
sir.
so Dr. Algo, just one thing I want to mention. So this submitted and map thing is different on this thing is that this study was done in mouse and we submitted protein IDs which are mouse IDs and we clicked project to humans. So these proteins are mouse proteins usually and they were then projected to humans and the human proteins were mapped in the pathway. So this is thing. So if this study is done in humans, you will see these things should be essentially the same. So that's one thing that can happen. I just need to cross check this and once more I will get back to you. Yeah, so any question? Yeah, Hina, go ahead, please.
Sir, entities FDR, what is that?
So there is a false discovery rate.
That FDR is the confidence, like how much confidence the entities you gained is good. So 1% FDR means 1 by 0.10. That means high echo is happening. Echo is happening.
Okay, just sorry.
So 1% FDR means high stringent criteria, high confidence. Okay, as you decrease the same, you will start getting low confidence or the low stringency criteria. But if there generally people do recommend for 1% FDR, that means what your entities is high confident and your data can be reported, can be communicated. Similarly, you can check if you are not going through FDR, you can check through the fold change. If you are not going through fold change, you can go through this one, p value.
Okay, sir. Got it.
More questions, and I hope then everyone should be comfortable with the reactome and the metaboanalyst. Maybe a detailed session of metaboanalyst and all the normalizing strategies can be done some other day, but this was to give you the basic flavor of that. So coming to the script questions, okay, coming to the script question, Sana, you have asked to see what proteins are present in a sample unknown. We do PRM, discovery based. PRM is not a discovery based, it is a targeted based. Discovery based can be a level free quantification, which can be done either in way of data independent acquisition, LFQ quantification, or we can do a SWAT MS, okay, sequential window analysis. So in all the ways you understand, like when you are doing a discovery, you just want to identify what all proteins are there in a protein detection range. Okay, Sana again. Hello, Sana. You heard what I was telling,
There's some
correct?
network issue.
Okay, so in the discovery stage, you want to identify what all proteins you are getting in the coverage. When you do a control versus disease, you identify which proteins are significant. When you do a fold change analysis, you get which proteins are upregulated and downregulated. And when you do a t-test analysis, you come to know which proteins are significant. Now you take both the proteins which are bold fold change passed and a t-test passed. These are called DEPs, differentially expressed significant proteins. Okay, so these are the proteins which we are interested in. Once you make the list, then you go for targeted verification. So targeted verification can be done with the help of many strategies such as SRM, MRM, and the PRM that will validate that these proteins, what you have got in your discovery space, is validated in the blinded cohort. Okay, now you asked how to develop a discovery data set library in the Orbitrap in a PRM. You take the MZML file, there is a procedure, you import that MZML file in the Skyline and you can generate a library through that. If you don't want to use MRM, your discovery, it is always advisable to use your own discovery studies. But if you want to directly go for the targeted verification and don't want to, and you cannot perform your discovery study, you can always integrate Prozit. Prozit is an algorithm which generates synthetic library, like it will go and cleave synthetically proteins and the repository on the basis of that it will generate a library. And you can know more about Prozit, how to integrate in the Skyline. The tutorials are already available. The maximum number of proteins that can be spotted, and what does it depend on? I couldn't get this question. How is fold change calculated? Fold change calculated by fold change can be calculated in two bases. One is the log fold change and one is the base fold change. Base fold change is by final outcome by the initial outcome, and the log fold change is logarithmic value of the log outcome by m. So if a log fold change is 1 or 4 means if a log fold change is of 1, if you convert that into the numeric value, it will be something around 4 or 3. Okay, log to the base 10 or log to the base 2, they be depending upon you. So if you see a paper which is instating the fold change or log fold change, please do look that very carefully. So I hope every question is answered to you, Sana.
Sir, one question. I'm sorry because of the network issue. I don't know. Maybe I couldn't. I mean, you said and I couldn't get it. But it was like in Skyline. How do we undo a deleted transition like during the refining?
Okay. So during the refining, you check the p values. Okay. The p values or the dot p values, you get it from the uh, library match. When it is having a match of the library, the dot p value, if you are getting 1 is very good. But if you are getting more than zero point 0.7 and 5, you get, if you are getting more than zero point 0.7, then it is can be considered. Those kind of peaks can be considered. You just click on the transition on the left hand side and you delete it.
And how do we undo it?
Undo it, you have to save, you have to save the data in a different format. So when you are refining your pics, you can label it as version 1, version 2, version 3. You can import your old pic file.
Okay, okay.
You are getting it
Okay, sir.
correct. When you are deleting some pics from your raw data
Mhm.
or the Skyline file, that is a different version. You can save it as refined version 1.
Okay.
You can always go backside and there is a provision, like if you tell that show all pics and imported pics, you can get the all pics also. Just remove the duplicate values in the Skyline. And second thing is that at the side of the transition, if you see a chromatographic mark, okay, that means what, that thing is coming in your global data also if you are using a global discovery map.
Okay.
We in that way we go for high confidence peptide. But
Okay.
always see that dot p value should come between 7 to 1, 0.7 to 1, 0.7 to 1.
Okay, sir. Okay. Thank you.
Any questions? I think after 5 days we will. Yeah, one question is from Hina Vidani. Yes, Hina.
Sir, can we do metaboanalyst for spectral processing? Like one demo session on that?
Spectral processing. See, MetaboAnalyst is just a software. Similarly, you can do it with other softwares also. ठीक है? There are a lot of softwares which are available online. I think one is from the broad analysis, that is Prodigy. Prodigy is also there. There is one another software. You have to understand the biostatistics of it. What is the sequential steps of data processing? Why you are doing normalization? Why you are doing transformation? Why you are doing scaling? Why you are doing multivariate analysis? So once you understand this, it is not only applicable only to the mass spec. You can apply it in cell culture also, NMR also, FTIR also, anything. Like if you understand the steps of this is called biostatistical analysis. If you understand that, you can apply it anything. This we have given you only for your practice so that you understand what are the steps involved. Okay, this is not only limited to the mass spec. You can apply it in your plant studies also, animal studies also, any studies also. You are getting it, Hina, correct?
Oh, yeah. Okay. So one more question. I'm working on snake
Yes.
venom, and currently it is very difficult to get venom. So I
Who
was
is that?
planning for
Talk to Sana. Sana is having lot of venom. Means she has worked on the snake venom.
okay, okay, sir. But still it is
Sana,
schedule A right now.
So yes, sir. It's
you have worked on the snake venom, na?
yeah, but unfortunately, it like how Vijay Lakshmi is saying is it is right that it is very difficult to get venom, and even when we get it, it's like hardly a few microliters.
Okay.
So yeah,
So
I
Hina,
think
you are also working on snake venom.
Yes, sir. I'm planning for proteomics as well as metabolomics from the patient samples who are bitten by snake. So that is
That
under
is a
process.
different Sisi,
Yeah,
those are
see
different
that you'll
aspect.
get you'll
yeah,
get from
yeah,
the patients.
yeah. That part is okay. It's under the process. But part one, I want venom and to do proteomics of venom. So is there any repository from where I can get this proteomics done from venom and analyze it?
You want to do the
See,
proteomics of your sample, or you want to already see what all is present, like the already done database?
My first objective
and
was to get venom and
switch
to
on the
see it.
camera. We can have a good discussion. Switch on the camera. Can have a
Yeah.
good scientific discussion.
So I was planning to get venom, but now it seems it is very difficult as the venom is under Schedule A. The snakes are being in Schedule A.
Correct.
It is very much difficult to get venom. So my main objective is to get venom and do proteomics or metagenomics of the venom as well. I was planning that. So
Yeah, because
what?
it does take a lot of time. You need particular licenses also
Yeah,
if you are working on snake venom. So, like before getting the venom, also there'll be some time where you'll have to devote to all that administrative kind of work.
yeah. We have signed an MOU with Gujarat, only Gujarat Forest Department, but still it is taking time. So meanwhile, I am planning the patient samples, their serum urine, go for proteomics and metabolomics to find the biomarker if possible.
And I think
And
to
I
work on
wanted to know one thing. I just
Yes.
wanted to know one thing. When it is in a human patient's body, it is not contagious, correct?
Yeah, it is not contagious.
Means anyone can handle those.
Yeah, anyone can, by skin contact, it is not that until and unless
No,
it
no.
is.
So what is the thing is that I know there might be few neurotoxins, correct?
Yeah,
yes,
During the
cardiotoxin.
time,
Yeah,
cardiotoxin, neurotoxin. So I hope
yeah,
if it does not require a special processing, you will inactivate anything which is harmful to the others before analysis, correct?
correct, correct. No, it is not the problem.
I think working from patient sample, if you want to see the proteome of venom in patient sample, will not be difficult, but directly venom will be difficult, and for that you have to even undergo training to
Yeah,
handle venom. Where are you
So
working?
Hina,
I'm
so
working at
Hina,
IT.
Sana is one of the important contact. You can talk her offline and she will guide you, huh?
Sure.
Okay. And you both are coming on 30th also. At that time
Yeah.
also you can plan the experiment. So we have some questions from Dr. Vijay Lakshmi also.
Hi sir, actually I just wanted to know, like Reactom, we have KEGG and Wiki pathways, right? So what is the difference, like how to choose right tool for our
See,
experiment?
these all are pathways. Some are cake pathways, like if you go and see their repository, some are maintained by the Japan, some are maintained by the US. So what is the major thing is that Some pathways are updated and some pathways are not updated. Like the cake pathways, and some hits you will get in other pathways which we may not get in another pathways. So that is why we go for a combination of pathways such as Wiki, KEGG, and PubMed, anything like which we'll get that will give a pure result. So we generally use Reactome because Reactome has the most updated form of the pathways rather than individually you go for KEGG and means other pathways, David and all those things. This is the most updated form Reactome and it is easy to use also. You can download the images also, very good images do come for the whole reaction means whole process machinery. You can show it and explain very well, which is not there in KEGG and David.
Sir, regarding protein-protein interactions, we will be having demo sessions on that topic, like using string and all. We do PPI analysis, right?
It is very easy. One day we can take on
Yeah.
demo means protein, cytoscape or string, anything. But you
Okay.
have to understand, like once you go through these pathways,
Mhm.
whatever important proteins you are getting, like for a particular pathway of infectious disease and all those diseases, these pathways will remain the same. Try to look how many proteins are coming in a pathway.
Mm.
Okay, based on that only you will do the string analysis, KNN networking, and you will find the clusters. So half of the answers come from the pathway analysis only. Once you get the answers from the pathway, then you do a PPI networking and see that among that pathway how many proteins are clustering. Okay, K means or whatever, like whatever clustering you want. And once that has been done, you can draw your own diagram, very lucrative diagram, that these three proteins will be affecting in these three regions, either mitochondria or that kind of, because you already have the information for the pathway that in this place these proteins are dysregulated.
Thank you.
Any more questions? Okay, great. So we will be very happy to meet you on the 30th. And let's see, like we are also eager to meet our first batch. And I hope you all are enjoying the lectures, huh? Because most of the requests are coming that I cannot maintain my this pace with the ongoing exams and all those things. We can understand there are a lot of ongoing exams, but do plan your timings very carefully because this is the one time learning what you will achieve, and it will help you to build strong base. Okay, and we will try our best to give you these things. So without further ado, we will be ending the session here only. The recording for the Metabo analyst will be available and we will be sharing it. But for industrial talks, the recording will not be available. If you have any questions, any more questions?
So
If
for
not, then
the
yes.
same data file that has been shared, we have to use the same now for the assignment.
No. Assignment data files are different. This is only for test. Then to
No, not
everybody will get.
the one which is shared earlier in the assignment
Yes.
Google
The
Drive
earlier
link.
one you have. The earlier one you have to use. No, so everybody will now get 10 out of 10.
I hope so.
Yeah, it is.
Yes.
Okay, great.
I
Have a
have one
happy
more
weekend.
question. I have one more question. Sorry, sorry. Trouble, last moment trouble. I have one doubt. Like Metabo Analyst version 5 or 6 we have to use for that assignment.
Any version you use the if you keep the parameters same, the result will be same only. 1 plus 1 always becomes 2.
Okay.
It
Okay,
will
sir.
not change to 3 or 4.
Yeah. Okay. Thank you.
Okay. Only the thing is that Metabo Analyst 5, it's any files, like if you go for the software upgrades and all those things, like this will give you the most advanced pictures. In Metabo Analyst 5, they downloaded the box plot and all those plot, the downloadable figures were not of that quality. But yesterday I was looking at Metabo Analyst 6, the pictures are very good as a publication quality image. So this will means this will suffice many of your supplementary figures. And at the end also, the Bhargya, have you shown like how to download the all the data which you have analyzed? At the end, there is a provision. Whatever analysis you have done, you can zip it and download on a single folder. So when you zip and download, you will get every box plot and everything there. Okay, great. Then we are ending this meeting. I am stopping the recording and have a happy weekend. See you.